is Bob, the Hawaiian bobtail squid, Euprimnuscolopes. During the day, he remains buried in the sands of shallow reefs, but upon the setting of the sun, this nocturnal animal emerges from the comfort and safety of his home to search for food.
But how does he protect himself from the dangers of the water and the sea of unknown? The key, his light organ is used to provide camouflage through counter-illumination to avoid casting a shadow on the ocean floor.
Bob owes his glow-in-the-dark powers to the bacteria living within him, Microbio fisheri. A fisheri is a gram-negative, rod-shaped bacteria that lives in symbiosis with various marine animals. Their colonization induces morphological changes within Bob's light organ, in which ciliated cells of the photophores selectively draw in the symbiotic bacteria. The light organs of the squid contain reflective plates that intensify and direct the light produced due to proteins known as reflectants. They regulate the light for camouflage, requiring the intensity to match that of the sea surface above to obscure Bob's silhouette.
Without these bacteria, Bob would be in grave danger. Predators would be able to recognize Bob's silhouette from below, and Bob would become dinner for a hungry seal. But how can one small bacteria create these luminescent properties? There's not just one singular bacteria within Bob, but hundreds that work together through a process called quorum sensing. Gene expression is regulated in response to fluctuations in cell population density. Each bacteria produces chemical signaling molecule called AHL, which is a type of autoinducer. These molecules are produced in response to increases in the density of Vibrio fischeri. The detection of a minimal threshold stimulatory concentration of an autoinducer leads to this alteration in gene expression.
Let's take a look at these processes on a deeper biological level. A fischeri's luminescent properties stem from the Lux operon, a luciferin-luciferase system encoded by a set of 5 genes, Lux C, D, A, B, E. Most importantly, genes Lux A and Lux B code for the protein subunits of the luciferase enzyme. Further, Lux C, D, and E code for a fatty acid reductase complex that makes the fatty acids necessary for the luciferase mechanism. The final luciferase protein produces blue-green light through the oxidation of a reduced flavin mononucleotide and a long-chain aldehyde by diatomic oxygen.
So how does Bob control when his light organ will shine? A fisheri responds to its surrounding environments. The Lux system acts amongst high cell density populations, producing significant levels of luminescence. This causes Bob's light organ to shine so he can safely find his dinner and return home happily with his stomach full.